So my research involves modelling the population dynamics of Nephrops in the Firth of Clyde. And Nephrops are a type of lobster, and they're the lobster that their tails are used to make scampi. So we're looking at modelling their population dynamics, and particularly taking into account infection by the parasitic dinoflagellate hematodinium. And this is a parasite that causes changes in the muscle tissue of Nephrops, and it ultimately leads to a change in the taste of the Nephrops. So far I've been looking at modelling the dispersal of Nephrops larvae in the Firth of Clyde. So we're using a hydrodynamic model to simulate how these larvae would be transported by the ocean currents. We need a high-resolution hydrodynamic model of the Clyde, and we also need to know whereabouts the Nephrops are, so where these larvae are being released, and then we just put these two together and see where the larvae end up. So Nephrops need muddy substrate so that they can dig their burrows. So we actually did some GAM modelling, so a generalised additive model, and this allowed us to predict the percentage of mud across the whole region. So we can predict mud in unsampled areas based on predictors like depth and distance from the coast and ocean current velocity. We compared that to actual fishing data of Nephrops trawlers in the Clyde, and we saw that these two matched up pretty closely. In terms of where did we put our release points, we just put them all over this patch of muddy sediment just to cover all options. The length of the larval stage is dependent on temperature, so we can take the temperature data out of the model, figure out when these larvae are going to reach adulthood, and then look at where they are at that specific point. The interesting thing was that the percentage of viable larvae increased throughout the hatching period, viable larvae being those which reach the point of metamorphosis over an area of suitable sediment. So we found that this increased throughout the hatching period and was highest at the end of June. And this was because seawater temperature increased, meaning that the length of the larval stage was shorter and there was less time for the larva to be swept away from the patch of suitable sediment. So some of the larvae released from the lower regions of the Clyde are swept right out of the Clyde, and some of them transferred to other patches of suitable sediment in the Sound of Jura and even in the Irish Sea. And there's possibly reversal as well, so some of the larvae from the Irish Sea could well be transferring back into the Clyde, so sort of mixing kind of effect. So I'm going to use this information to incorporate this into a population model of Nephrops in the Clyde. So we're going to create a spatial population model. So in order to do that, we need to know how many individuals are transferring between spatial cells in the model. So once we've created our model, we will run it under different scenarios, so we could look at what would happen if we changed discard policies, or what would happen if we changed fishing intensity in certain areas. We could look at the yield curves of Nephrops under different scenarios.